Welcome everyone to the final of our evening monkeypox webinar series. This is a partnership between the STD HIV Prevention Training Center at Johns Hopkins and the Maryland Department of Health. My name is Barbara Wilgus and I am the program administrator for the Prevention Training Center. And I want to welcome you today to a fabulous lecture. I'm very excited that you're here. A few housekeeping instructions. Your Zoom registration to enter the webinar. So when you entered your name and email to get your Zoom link, that's your sign in for the day. And that's how we're taking attendance. So if by chance you're using a different email for Zoom than the one that you use to register for the course, please email Bambi Hamby at mhamby1@jhmi.edu with this information. Similarly, if you're watching in a group, please email Bambi the names of the watchers in the group that aren't on the Zoom screen, because for CE credit, we need to count every person who has attended as having attended. This is a webinar format, so you probably have already noticed that you do not have access to your camera or mic. So if you have a general comment or a technical question, please enter it in the chat box below. If you have a question for our speaker, please enter it in the Q&A box below, and we do anticipate having time for questions. These webinars are being recorded. Recordings will be accessible via the Maryland Department of Health MPX webpage under the section Information for Clinicians. The MDH MPX webpage is located at health.maryland.gov/monkeypox. To receive continuing education, you must have registered for this course via the NNPTC. You must be here attending the live and interactive viewing of the course. Again, we are taking attendance via your Zoom registration. Unfortunately, if you're viewing this as an archived recording, this does not qualify as CE. For those who have attended tonight, we will email instructions for how to get your CE credits following the course completion to the email address with which you registered for the course itself. You're going to have 30 days from today to go to the CDC website to get your CE certificate. It is a process. There are several steps. So if you have questions, make sure you're getting all the way through to get your CE certificate. If you have questions or concerns, you can email Jean Hoover, jhoover6@jhmi.edu. And again, CE credit is only offered for the live and interactive version viewing of this webinar. A few words about the national network of STD clinical prevention training centers, of which we are one. Here's a map of the US and all of its territories. And the NNPTC is actually a national network. We are the region that you can see in teal there. But if you're hailing from another state or territory, find your state, and there is a prevention training center regionally for you, providing technical assistance and training right in your geographic catchment area. Some national offerings that we have. If you are a clinician and you have an STD question or consultation that you need an expert opinion, you can go to stdccn.org, submit your question, need for technical assistance, consultation. It'll be routed to a subject matter expert in your region. So if you're here, it's going to go to my bosses. And you can expect a response within 1 to 5 business days, depending on the urgency that you have set. So this is a wonderful free resource for clinicians in our area. Another national offering is the National STD Curriculum. They have updated it for the 2021 CDC STI treatment guidelines. This is a free, up-to-date educational website with well over 30 free CME credits. There's a self-study, go at your own pace, CE online ongoing education, 20+ question banks, a wonderful podcast with STI related topics of all kinds, and a group learning tool where you can assign units, track your members' progress. So that's all located at www.std.uw.edu. We are partnering with the Maryland Department of Health for these evening webinars, and I do want to remind you again that many resources, including these recordings, will be available on the MDH website, health.maryland.gov/monkeypox. There are up-to-date, clinician letters, information for providers, but also a host of patient handouts and things that you can give your patients that you're seeing as well. And really importantly, since we're talking about vaccines tonight, this is where you can find the Maryland Department of Health statewide MPX vaccine pre-registration system. This is an ongoing effort by the Maryland Department of Health to provide MPX vaccine across the entire state in an equitable fashion. And it is really the best way to get your patients in line and ready to get their vaccines, which is highly important, as you will hear. And finally, from our, of course, partners at the CDC, the STI treatment guideline mobile app is now available for iPhone and Android devices. So you can download using this screenshot, or you can just go to cdc.gov, this QR code, or go to cdc.gov. This is more comprehensive now. They've expanded all kinds of offerings, including a section for taking a sexual history, some clinical tools, and you can actually access the STD CCN directly through the app now. So all of it's available right in the pocket of your lab coat. So those are some other amazing resources ready for you. And I am going to stop sharing my screen now. And with that, it gives me great pleasure to introduce our speaker for tonight, Dr. Noreen Hynes. Dr. Hynes is an associate professor of medicine in the Division of Infectious Diseases at Johns Hopkins and the Division of International Health at the Bloomberg School of Public Health. She serves as the associate medical director and research director of the Johns Hopkins Hospital Biocontainment Unit and the co-director of the JH-MPH Infectious Disease Concentration. She is a renowned subject matter expert in tropical medicine and tropical diseases, high consequence pathogens, vaccines, including vaccine clinical trials for high consequence pathogens, the FDA animal rule, and emergency use authorization. And you are in for a treat because Dr. Hynes is going to discuss everything you ever wanted to know about the MPX vaccine, and you're up for a fascinating time. So you can share your slides whenever you're ready and take it away. Oh, and unmute yourself.
I did. I shared my screen.
Oh, I am not seeing it yet. Maybe try again.
Okay, I'll end my slideshow. And I'll say again, share my screen.
There we go. There we go.
Okay, now I have to change this, whatever, to the type of screen show it is.
Perfect.
There you go. Okay, so it's a pleasure to be here. I'm excited to do this. I know I'm the last, so hopefully you're already well informed by many of the previous speakers. And so I'm going to speak very specifically about monkeypox vaccines and the science of them and their use, and just a little bit about the operational realities, because when I've given this talk before, we had someone from the Maryland Department of Health deal with many of those things that you can find on their website and on the app. Okay, so what I hope to accomplish during this brief time we're going to spend together is to sort of explain to you what even led to the development of this modified Vaccinia Ankara Bavaria Nordic, which is the real name of the unit Yos, which is the brand name for this vaccine. It has a different name in Europe. And then talk about the similarities and differences between the ACAM 2000, which is actually a vaccine licensed for smallpox only, and Unios, which is licensed for the use in the prevention of both smallpox and monkeypox. And then compare the provisions of a licensed vaccine to one being used under an EUA, because as you know, there are certain provisions for the use of Unios which only allow it to be used under an emergency use authorization, rather than as a licensed product, and then discuss the efficacy and immunogenicity of this vaccine for the use against monkeypox virus. So as you heard from Kelly Gibo, who spoke to you earlier, she gave you this wonderful one-slide vaccine summary showing the difference between the two vaccines, which both could be used for the prevention of monkeypox. The Unios vaccine and the ACAM2000, you can see they're both live, but the thing about ACAM2000, it replicates, whereas the Unios vaccine doesn't. And you can see differences in doses, 1 versus 2 for Unios, and you can see the different approval ages, the data on efficacy, what it's contraindicated in. Of course, there are many fewer contraindications for the Unios vaccine, and it's safe to administer to persons living with HIV, atopic dermatitis, et cetera, and immunocompromised persons. So the vaccine that we're going to be spending most of the time talking about, but I think it's important to understand why ACAM2000 could also be used. So I'm going to take you through a brief trek in history, because I think that understanding where these vaccines came from certainly will help you understand why they can be used now, and maybe you won't be so skittish about them. So, you know, in the beginning, there was smallpox. And as you know, it's a unique human pathogen, variola-virus. It only occurs in humans. It's not like monkeypox. Monkeypox is a zoonotic infection, which occurs in animals and then comes into humans, but can be also transmitted human to human, as we've seen in the current large global outbreak. And it was at one time a major killer, and a decision was made that it was time to eradicate it, because the characteristic that it only occurs in humans made it a candidate to be eradicated. So vaccines actually existed for many years. But first, Jenner noticed that the milkmaids who got cowpox didn't get smallpox. So there are many viruses that end in pox. There's rabbit pox and mouse pox and monkeypox and smallpox, which is of course human pox virus. And at the time, way back in 1967, WHO did a survey and found that there were 59 labs making vaccine using vaccinia virus, that is vacca from the name cow. But you should know that although we say that vaccinia virus is cowpox, there in many places it had some other viruses in it as well, maybe some mule pox, some horse pox, et cetera, in a mix over time, with clinical efficacy against, importantly, orthopox virus. So safe and effective against smallpox, monkeypox, cowpox, horsepox. So you can see there was a wide range of things the vaccinia virus would protect against. And so at the time of this survey back in the 1960s, there were four dominant groups. There was this Lister Elstree strain that was used mostly in the UK and its colonies, that was made in 39% of labs. The NYCBH, which is the New York Board of Health strain, which was used in 12% of labs that were making vaccine, the Paris strain and other strains. I think it's important for you to realize that in the early days, vaccines were not just made in labs, they were often made in health department labs. So the anthrax vaccine that was made way back in the days of Wilsorter's disease here in the United States was made at the Michigan Department of Health, and smallpox vaccine made at the New York Board of Health. So because of all prevention, et cetera. And if you take a look at places where there are vaccine research centers in academic settings, they are invariably in schools of public health rather than in schools of medicine. And this follows along with the longstanding tradition that vaccines were part of public health. So as you can see on the left, the New York Board of Health strain was made by the manufacturer Wyeth under the name Dryvax. That was the common vaccine that was used here in the US. So this smallpox vaccine from these 59 labs, so how did they even make them? How did you get to the point where you had seed virus? That is, they were like the starters. For those of you who make yogurt and breads and things, you have to have your starter culture. Well, this is where did they get the starter cultures from? So imagine this. 51% came from the skin of calves. They inoculated calves, and then they would scrape the lesions and then clean it up the best they could, grow it, and then it was made into vaccine over time. And then 6 were from other bovines. We had 3 that were from the chorioallantoic membrane of chick embryos, and 3 from tissue culture from bovine embryonic fibroblasts. And the biological features in terms of how high was the viremia you would get, how big was the take or the lesion at the site of the introduction of the vaccine, all had to do with how many times it was passaged in cells before it became the vaccine. So as I noted, the world decided, that great, wonderful we, decided we were going to eradicate this. And the goal, because that became a goal, WHO decided, well, we better have some standards for what the vaccine is that everyone is going to use. And you're going to see politics has been, politics is in public health and always has been. So you can see the Lister Elstree strain from the UK was used, the New York City Board of Health strain was used. But as you can see, this EM63 wasn't on that list. It was in that other classification, but it was made in the USSR. And so it was decided it was quite an adequate strain. And so these 3 vaccines were considered to be what we wanted to use for smallpox eradication globally. It was considered to be highly protective within 3 to 4 days of receiving it, of an exposure. If you got this within 3 to 4 days, chances are you did not become symptomatic with smallpox. But we learned pretty early when we made this a routine vaccination that if you were very young, and before the 1950s, we gave this to newborns and wound up with very, very severe adverse effects, SAEs. But then once they changed to giving it to small children older than the age of 1, then there was a great decrease in these serious adverse events, or SAEs. But to give you some idea of what some of these severe SAEs were, in 1947, in New York, there was a New York City outbreak after a traveler from Mexico who had smallpox entered, and there were 12 cases in New York City and 2 deaths, okay? But think about this. The New York City Health Department was able to respond very, very quickly and gave 6 million doses of vaccine in 1 month and had 6 deaths among the 6 million, or 1 death per million doses given. And then in 1968, I'll show you more data in a little bit about this. Surveys were done here in the United States in 10 states through the CDC. And they found that when the vaccine was given, this post-vaccinia encephalitis, they found was in 3 to 12 per million, but realized it was not actually associated with the virus multiplying or replicating, but was actually probably an immunological phenomenon, much more common in women than men, for example. And this is true with other live vaccines, such as yellow fever vaccine, where a similar meningoencephalitis is seen more commonly in women than it is in males. And then progressive vaccinia, 1 to 2 per million. And in the upper slide, you'll see how that looks like a really nasty looking wound that's spreading out. And that's progressive vaccinia. It just keeps going and going and going and going. And 1 to 2 per million, usually seen in persons who are immunocompromised. Then there's eczema vaccinatum shown in the lower photo, in which someone with eczema, if they got their vaccine, let's say in their arm, and they touched that, and touched an area where they had eczematous lesions, they could develop this really serious illness in 10 to 40 per million, so quite a few. So during smallpox eradication efforts, it's very important to realize way back then, we first knew of monkeypox in humans. Back in the 1950s, we knew it could occur in monkeys because it was found in caged animals up in Norway, where they were trying to study smallpox, but non-human primates don't have the same manifestations that you see in humans. And so it was problematic using them to study smallpox. And then they discovered it in the 1970s and realized that it wasn't the same as smallpox, but it was prevented by using smallpox vaccine. Remember, orthopox viruses, they're both in that same family. So then, you know that the Unios vaccine is this modified Vaccinia Ankara. So where did this all start? Well, way back in 1953, before we had eradicated smallpox and declared it eradicated in 1980, Ankara in Turkey turned over their strain of smallpox and gave that strain to the Institute for Infectious Diseases and Tropical Medicine in Munich. And then they cultivated it on egg, chorionic allantoic membranes, and then it became chorioallantoic vaccinia virus Ankara because they wanted to give credit to where the virus came from. Then they took that, just like other vaccinia viruses, and then grew it again on the skin of calves to make a smallpox vaccination to be used in a campaign in Germany from 1954 to 1958. But then in 1958, they noted that there was decreased cell tropism. That is, it didn't grow as well in cells as they were passaging it more and more and more up to passage 371 in primary chicken embryo fibroblasts. Then when they further passaged it by 1968, they noted it now had even more tissue tropism. It had greater difficulty growing in different animal species cells. And they renamed the strain now modified Vaccinia Ankara. And a small number of humans received the vaccine to see what would happen. And they didn't get a usual take like they got from smallpox vaccine, which gives you this big, juicy looking item where they inject your vaccine. So then the institute in Munich gave to the Bavarian State Institute for Vaccination, they gave them this strain to test for a suitability to be a smallpox vaccine. And so they took passage 517. You see, it came as passage 0 when it came as vaccine virus from Ankara, Turkey. And now it's gone through 517 passages. And they wanted to use it in Europe to test. And they did eventually use it on 120,000 people, which ultimately I'll talk a little bit about. And then they decided that how it potentially could work well was to give one dose that didn't seem to give a big take, but seemed to do something as a prime dose, and then boost with the live replicating strain. But they also had looked at in rabbits and non-human primates that it just didn't give the typical lesions that you would have seen with the classic vaccinia or smallpox vaccine virus. And so these are what the clinical trial showed. These were done before smallpox was eradicated. So that's an important thing to know. They used a dose of 2.10 to the 5th international units. That was in 0.2 milliliters of solution. They gave 107 people. They got no pock lesions, which is the classic take, a single big pock, juicy pock, for any of you who've received smallpox vaccine. live attenuated smallpox vaccine. They didn't get any fever, and they had no classic antibody findings that you would have seen when you use that Lister-Elstree strain from the UK. But when they got their second vaccination with the Lister strain, they did get a typical follow-up vaccination take, but not the first primary take like you would see when you got the vaccine the first time. So they were encouraged. And then 7,000 more people were immunized with similar findings. Then it was transferred to the Bavarian State Vaccine Institute in Munich. And as you can see, it actually in 1977 received marketing authorization for this to be the pre-vaccine against smallpox in Germany as the first dose of a 2-dose regimen, with the second dose being a live attenuated strain. But as no clinical efficacy data was present because there was no smallpox in Germany by that time. The only place there was still smallpox was in the global south. So then the question is, do we have what are called immune correlates of protection to replicating vaccinia viruses? So the question is do we know if there's a level of antibody that you can reach that ensures you or assures you that you're protected against smallpox? We don't know what that level is, but we do know that after a percutaneous standard dose using what's called the bifurcated needle, greater than 95% of people do produce a neutralizing antibody of greater than or equal to 1 to 10. But also we've come to realize that those people who get the live replicating smallpox vaccine, Dryvax and all the other ones, if you had a take that, and it looks like a pustule, it correlates with protection. And so this antibody level of greater than or equal to 1 to 10 persists among 75% of people for 10 years after a second dose, and less than or equal to 30 years after a third dose. Because one of the questions we often hear about different vaccines, particularly when they're live, is this durable immunity for the rest of one's life? So it appeared from what we knew from smallpox eradication that it wasn't sterilizing immunity for life. So the second dose of vaccine, less than 10% of people with neutralizing antibody of greater than or equal to this magical 1 to 10 before they got immunized had a typical primary take. They had sort of a secondary take. It was just a little bit of something. And greater than 30% have it if their titer is less than this 1 to 10. So there was something almost magical about that special neutralizing antibody titer. And so persons going to work, however, you would say, okay, so you could do it once every 10 years. But they made the recommendation at that time that if you're going to work in a smallpox endemic country, then you should be vaccinated every 3 years. And that's what was done prior to eradication. If you were going to work in smallpox countries, you got the vaccine every 3 years. And so even after eradication occurred, there were so many still unanswered questions. And this big book that was written by D.A. Henderson and others, D.A. Henderson, the former dean at the School of Public Health, many of these questions still exist today. Can we develop a safer smallpox vaccine? And I'm going to tell you a bit more about Junios. What is protective immunity? We still aren't sure about that one. And how long does immunity last? We don't know really that one either. So what happened to MVA as we got smarter and were better at looking at the genetic features of viruses? As we improved our modalities in science, we came to realize that the change from all the other vaccinias to including vaccinia, the original vaccinia Ankara, to the modified vaccinia Ankara, it lost 15% of its parent genes. So, wow, that's a lot to lose, but what did it lose? And what importance were those losses? So the genes regulating host range changed. So you couldn't infect all these different animals like you could with vaccinia virus. So you could infect fewer types of animals and fewer cell types. And we found that was very restricted in mammalian cells, and it had no productive replication in humans. They could have an immune response. You could even get protection of some sort, but it didn't replicate, which was important when you think about it for someone who is immunocompromised. So the hallmark of vaccinia virulence in the laboratory was to take regular vaccinia vaccine and inject it into the brain of mice, and it would be neurovirulent. But when you did that, even at very high doses with MVA of the modified vaccinia Ankara, they didn't even get sick. So we knew that there was something going on. And in the cartoon on the right, you'll see the virus at the top, it's entering, and then it's getting uncoated, and it does everything, but it doesn't leave the cell again replicating. So it does all the right things. The body sees it, it recognizes, it has vaccinia proteins, et cetera, against which it can create an immune response, but it doesn't release it to replicate. So what's its safety profile look like in animals? So in immunocompromised animal models, you can give an exceedingly high dose of this MVA. In mice, they don't get ill, the virus doesn't multiply, but importantly, if you waited a while, they actually were protected against the Lister estuary strain. So it wasn't as quick as the protection you would get if you were immunizing someone with a live replicating strain, but eventually they in fact did and were protected. And so immunocompromised rhesus monkeys, we're getting closer and closer to humans now, had similar findings. So we wanted to know more over time. What exactly were the loss of the genes other than having to do with viral replication? Well, we found that some of the genes that code vaccinia and other pox viruses with these soluble inhibitors for the interferons, like interferon gamma and interferon alpha and beta and tumor necrosis factor alpha, were also lost. So that meant that you couldn't have a cytokine storm with this like you could have with vaccinia, which again would be very important in a person who is immunocompromised. And then the structural genes, however, that encode for presenting to the immune system the fact that you have a foreign protein so it can make antibodies, and specifically neutralizing antibodies against that particular epitope or protein signature, actually didn't go away. So this was sort of cool when you think of all those passages, and it selected these things to lose and other things to keep. And then routes other than by scarification, that is using the bifurcated needle, you didn't need to use, would be just as effective. So a single intramuscular dose in non-human primates attenuated illness, and 2 doses protected them. So that's where we were with MVA, all this stuff that they were doing in the background for years and years, even during the smallpox era. But then, there was no smallpox, no one was doing any research. And then we had 911, with the planes into the buildings and then the anthrax letters. It wasn't that people weren't discussing, could we have a terrorist event? But this sort of put everyone's concern on steroids. So they took a look at what we had in what was then called the National Pharmaceutical Stockpile. And we did have Dryvax. Remember, this is the smallpox vaccine made by Wyeth that was in the and maintained in the National Pharmaceutical Stockpile. But there certainly wasn't enough for everyone in the whole US. And so there were maybe 20 or 30,000 doses, and we knew maybe we could dilute it a bit. So it was still licensed. It was in this stockpile. It couldn't be commercially bought because the US government owned all of the doses. And there are a whole bunch of contraindications, because you remember from that early slide I showed you from Kelly Jibo's talk that there were many reasons, and a lot of people who couldn't get the live attenuated vaccine. And so the decision was we would only give it on a threat-based staged plan of use. So, you know, the military would get it, and then people who were caring for people who potentially were going to be exposed in hospitals, et cetera. So, but then over time, we then created a vaccine from the original vaccine. What was done is the Dryvax was grown, and then all the little plaques that were on the dish that was used to grow it, they've picked some of the individual plaques or colonies, grew it, selected one of them, and then grew that in a great amount. And then this is what ultimately became ACAM 2000, which is derivative of the Dryvax vaccine. And there is now enough of it in this, now what's called the Strategic National Stockpile, to protect 100% of the population of the US plus others. It has the same side effects and severe side effect profile as was seen in Dryvax, but because we looked much more closely when we were doing the human clinical trials, we also found that it causes myocarditis in 5.7 per thousand primary vaccinees. And the same bifurcated needle that's shown over here is used to administer it. But what you should know about this, the way this was licensed is by a thing called equivalence. Remember that greater than or equal to 1 to 10 neutralizing antibody? So it was put into clinical trials to make sure it had equal safety side by side with Dryvax, and then also was looked to see if it had equal neutralizing capability and the same take rate in the arms where it was usually given. So this was a vaccine that was, there was of course no smallpox, so you couldn't do an efficacy trial, but it was licensed under the purview of what's called equivalence. So, but it has the same side effects as Dryvax. So eczema vaccinatum, here's a case on the left, generalized vaccinia in a healthy child, and then this progressive vaccinia in an immunocompromised child. And I alluded to before that there was a 10-state survey done looking very carefully at the side effects from Dryvax back in the, that was done by CDC. Mike Lane at the CDC was the lead investigator. And as you can see, primary vaccination rates, and then on the lower part on the left is revaccination. You can see that inadvertent inoculation, so that is you scratch or take, and then you accidentally, give yourself another inoculation by scratching yourself. Generalized vaccinia, as you saw in that small child, because it just goes everywhere because the child became viremic. Then eczema vaccinatum, again, because the child had eczema, or even an adult this can happen to progressive vaccinia in immunocompromised persons, and this post-vaccinal encephalitis that can develop. As you can see, much more common with primary vaccination, but seeing that we don't give the vaccine to those under the age of 1 anymore, so you wouldn't get revaccinated at the age of 1, there was no problem there. But as you can see, still there were impressive numbers even on secondary vaccination per million. So what about this frequency of revaccination with smallpox vaccine? Okay, our data is based on data collected from persons working with not highly attenuated vaccinia strains. Okay, so they're working with essentially vaccine strains. They were getting revaccinated every 10 years, but to ensure that there was going to be an increased level of protection, more virulent non-variola orthopox viruses such as monkeypox got it every 3 years. What you should know, on the original label for that Dryvax made by Wyeth, when FDA issued the label, it was for the protection against and prevention of all orthopox viruses. It wasn't specific for smallpox. Okay, that's something important to realize. It was on label for all, which meant that even if there was, even the ACIP or its equivalent at the time, if they said it was for smallpox, then that was the indication. Once it was licensed and you could purchase it, if the label said it was good for any orthopox virus, you could certainly, as a provider, give it to a person for prevention of any orthopox virus, because you could commercially purchase it. That's not true if you have to get it from the US government. But then nature stepped in and helped us look carefully at whether or not this ACAM 2000 would work against smallpox. And so we had a monkeypox outbreak in 2003 in 6 states, shown here in this colorful histogram, and shows you a nice monkeypox POC on the left. And on average, the incubation period was about 12 days, but you can see it was very short in some of the cases, 1 and as long as 31. And 35 of 71 individuals had laboratory confirmed cases with all these different ways that they used to confirm it. And this is very reminiscent of all the ways people are being told now where to take your samples from for monkeypox. So here it was PCR, culture, immunohistochemistry, and electron micrographs as well. But 30 of the 71 people had gotten smallpox vaccine. 7 got it pre-exposure. 23 got it post-exposure. And monkeypox was seen in 3 of them within 2 weeks, but only 1 of 3 of them had confirmed monkeypox. So the other 2 had something else. But there were no adverse effects in this group, but that's very small, okay? So there was an animal traceback that was done, and it was an Illinois supplier to all sorts of animal pet shops in several places in the US. He had imported Gambian giant rats from Ghana. So then what you're often hearing, and you might be hearing, that this Unios vaccine only has 80% protection. Okay, smallpox vaccine only has 80% protection against monkeypox. And so this was a study that was published in 1988. So I want to bring something up. This is 8 years after the eradication of smallpox. If you take a look, there were people who, what they did was they looked at the secondary attack rate, that is the attack rate in people who were the contacts of index cases or co-primaries, and they looked at them very specifically according to what their smallpox vaccination status was and their place of residence of the close contacts. So if you look at this, it was the same residence as the primary or co-primary, other residents, and all the contacts. If you look at the unvaccinated, you'll see, well, their attack rate appeared to be much higher than individuals who were in the vaccinated group. But if you look at these, it makes it look like the protection of vaccination is only about 80%. But what they failed to ask as a question, and people failed to remember, during smallpox eradication, when initially there were not lots and lots of vaccine, they decided it was most important when they identified a case of smallpox to immunize immediately those who had no evidence of ever having been vaccinated. So there are more than a few of these people who could have been in this study who had been vaccinated years before smallpox was eradicated. So maybe even 20 or 30 years before. So this notion that smallpox only protected 80% against monkeypox is probably a misrepresentation of reality. And so this is a underestimate of protection at best. So what about the MVA vaccine or Unios vaccine that now sits in the Strategic National Stockpile? How did we get that? Well, it was estimated at the time when everyone was in a flurry of, oh, we need to be able to have enough vaccine in the stockpile to immunize every man, woman, and child in the United States. Oh, over 30 million doses. But then people said, but what about all the people who can't receive the vaccine? And at that time, it was estimated that about 30 million Americans could not receive a live replicating vaccine in the event of an attack against variola virus. And so in June of 2007, based upon the data that already existed for MVA, the Department of Health and Human Services signed a contract for up to $1.6 billion with Bavarian Nordic to conduct even more studies and to do further development of their existing MVA, what was called Imvamune, and it is MVA Bavarian Nordic, it's the same vaccine, and to deliver 20 million doses and up to 60 million doses to the Strategic National Stockpile. So it was to have some doses that were already vialed, and then some that would sit in bulk that could be vialed in the case of an emergency in a process called fill and finish. And so it was specifically going to target small children who wouldn't do so well with the replicating vaccine, the elderly, HIV, and other immunocompromised persons. And the first million doses came into the Strategic National Stockpile in July of 2010. So we've had it for a while. So then the question is, what are the adverse reactions that you can see in healthy adults? Okay, so the severe adverse events were pooled in over 22 studies that were carried out by Bavarian Nordic. In 4 of these, there were 4 severe adverse effects for which the vaccine couldn't be eliminated. That doesn't mean it really was, it's just that you couldn't eliminate it. For those of you who have done clinical trials, you know that an SAE doesn't mean it absolutely is a severe event. It's just that you can't rule it out, so it gets listed. So Crohn's disease, sarcoidosis, extraocular paresis, and a throat tightness. And then there were events of interest. And of course, the US was very interested to know if we were going to see that same myocarditis that was seen with the ACAM2000 live replicating vaccine. And there were there any cardiac symptoms in the over 22 studies? And there were 6 causally related, but none of them were severe adverse effects. So no myocarditis. So there were things like high heart rate, tachycardia, and palpitations, and EKG changes with a T wave inversion, ST segment elevation, and abnormal T waves, but none of them considered serious. So as you can see on the left, as you could imagine, there would be, as you get with many immunizations, pain, redness, swelling, induration, and sometimes itching at the site. There are a few vaccines that cause itching at the site, but it's not a rare event with some vaccines. And then, of course, systemically, many people can feel myalgias with this and other vaccines: headache, fatigue, nausea, and chills. So there were 22 studies that combined all these data, and only 42 vaccinees, only 42 of the experienced vaccinees, people who had gotten the vaccine previously, were 65 to 80 years old. So there wasn't a whole lot you could say about this age group because they were smallpox naive and previous smallpox. And as you take a look at these AEs, you'll see that the injection site often had more impressive side effects than someone who had seen smallpox antigen before. So what about, remember that greater than or equal to 10 neutralizing antibody level. So that what about the immune response to the available smallpox vaccines, comparing Unios with ACAM2000 and looking at their vaccinia neutralizing antibody, pre-vaccination and post-vaccination. So as you can see, for Unios, pre-vaccination was really no different than it was for ACAM before anyone got a vaccine, but post-vaccination. But the interesting thing to remember is that post-vaccination means 2 different things. For ACAM2000, there's only 1 dose. So post-vaccination for that has to do with 2 weeks after you got your second dose, whereas you had to get 2 doses of Unios, so you were waiting for a longer period of time. And as you can see, Unios, you had a much higher neutralizing antibody level. But remember, you primed them with a single dose. And there is some data from Bavarian Nordic, who has also done trials in Central Africa, in the DRC, and in West Africa, where they naturally have naturally occurring monkeypox, not like what we're seeing in the global outbreak. And they saw a great diminution in cases after just 1 dose of vaccine. So what about this, this licensed MVA Bavarian Nordic Unios vaccine that we now are offering to individuals who either have been exposed or at risk of being exposed to smallpox in the current large public health emergency of international concern that was declared by WHO. So the indication is you want to prevent smallpox and monkeypox. That's what's on the label for this vaccine in adults 18 years of age and older who are determined to be at risk for either of these infections. And this was initially licensed in September of 2019, long before this ever occurred. And the dosage of administration is 2 doses, a half cc on each, separated by 4 weeks. Okay, so that's important. And all of these, it can have some residual host cell DNA, can have some protein, benzonase, gentamicin, ciprofloxacin. So as you can imagine, if you're allergic to gentamicin or ciprofloxacin, this might be a problematic vaccine, but there's no preservatives and no latex stoppers. And then because it's a licensed vaccine, it gets a vaccine information statement, which is nice to be able to give people getting it in the licensed format, because it tells them why they are getting the vaccine, what's this vaccine they're getting, what to talk to their healthcare provider about, and the reactions they can get, and what if there's a serious problem. Additionally, there is a countermeasure injury prevention program, because this is considered a consequence pathogen. And so therefore, there are different types of injury compensation that one can get from the US government rather than from the manufacturer with this particular vaccine. But then the decision is which to use. And that's what came up early on in, should we be offering people the ACAM 2000 because we have so much of it, or should we be offering Unios, of which we didn't have very much? And it seemed like the histogram or the case numbers were soaring up. And the question was, if you had to give 2 doses of anything, you didn't have 1 million doses, you had 500,000 doses. You had enough for 500,000 people. And was that going to be adequate? So I think it's important to remember that there are very specific distinctions between the 2 vaccines, but the most important is that the risk for serious adverse events are yes for ACAM 2000 and no significant events during clinical trials for the Unios vaccine. The unfortunate thing is it needs 2 subcutaneous doses. And of course, it is replication incompetent, it's replication deficient, the Unios, whereas that's not true for ACAM 2000. And as I outlined think of the contraindications. If you take a look at the contraindications for the live replicating ACAM 2000 vaccine, you will notice that even if you yourself have no contraindications, if you're living with someone who does, such as someone who has eczema, then you would be contraindicated from getting the replicating vaccine because you could pass it to others in your household. So what about the contraindications for Unios? As you look for primary vaccination for ACAM 2000 and revaccination, there are yeses for just about everything on that list, okay? On revaccination of ACAM, you don't have to worry about 3 or more known major risk factors for cardiac disease if you're revaccinated. But look at, and If you're a household contact, there are a whole bunch of things you have to worry about. Pregnancy, there's someone in your household less than a year, etc., as I brought up. But there's only one yes for all the contraindications for Unios, and that is if you have a serious vaccine component allergy. That's it. So as you can see, this is why the decision was made that Unios over ACAM 2000 would be used. So the licensed product versus FDA has a licensed product versus what the ACIP recommends. So the FDA label for Unios gives you the indications for use. The ACIP tells you the recommendations and is considered legally as the guidance for the vaccine's use. But the caveat is, if a licensed product is purchasable, is commercially available, as long as you tell your patient what you're going to use it for or how you're going to administer it, and they agree to it, you can give it. But you cannot do that with something 100% owned by the US government who's then giving it to you. Then you must give it exactly according to the ACIP standard. And then anything else, there's no off-label use. Any quote unquote off-label use would have to be under an EUA. And that's why the intradermal administration is under an emergency use authorization. If you could purchase this, you wouldn't have to do it under an EUA. It would be a licensed product. So the interesting thing about the EUA fact sheet that's out there, it's really only adult adverse effects are listed because we don't have a lot of information for children. And you are required, however, to report to the vaccine adverse events reporting system any adverse events that you see, and you're also asked to tell Bavarian Nordic what you have, any severe adverse events, and also any vaccination administration errors. So what are the items under for using Unios under an EUA? If you're going to use it in children, you have to use it as it was made, that is 2 doses 4 weeks apart that would be given subcutaneously as a half a cc for each dose. And that's what the EUA is. It allows you to use it on those who are younger. But those who are 18 years of age and older, because of a potential shortage of the vaccine, you can also give it intradermally. You can give it intradermally because there are many vaccines that we have given intradermally with a very good immune response and no difference, both live and non-live vaccines. So we have given yellow fever vaccine as intradermal doses to decrease to increase the number of vaccine doses we had during global yellow fever vaccine shortages. We did it and did a study during the time when we were trying to, when we didn't have ACAM 2000 and we only had some dry vax left. We also did a study to see whether or not we could extend the number of doses we could get, and we could by giving them intradermally and even giving what are called fractional doses. And then we've also given rabies vaccine intradermally with a very good response, remembering that there are dendritic cells in the skin that rapidly take up antigen and present it to the immune system. So the ID can only be given to those who are considered adults. So people often ask, well, what about this ID dosing? Do we know anything about the adverse effects? Well, we know that we don't have a lot of data. There was one publication that came out of Harvard back in 2010, where they did note that if you gave it intradermally, there was a more pronounced local reaction with intradermal than sub-Q dosing. And that's been seen with other vaccines as well, that the intradermal immunization with MVA certainly allows for protein sparing. But they found that the magnitude and kinetics of intradermal IM sub-Q, the kinetics of those were similar, okay? But similar, but a little lower. So that's something to be aware of. But the thing I should tell you, and thanks to one of our providers at the person who's the medical director at the Baltimore City STD and HIV clinics, Liz Gilliams, was kind enough to provide this quick case history that I thought you would all like to see for a patient that she took care of, a 27-year-old man living with AIDS on antiretroviral therapy with a undetectable viral load and a good CD4 count, received Hynes vaccine based on his sexual behavior, but he had no known monkeypox exposure. He got his first dose subcutaneous, but his second dose he got intradermally 80 days later, so not the recommended 2 weeks later. So as you remember from that one study that showed you could really mount your immune response over time, remember, and that it could be that a delay also accounts for a more vigorous immune response to a second dose. He had no vaccine reaction history, but had some drug allergies. So at 36 hours following dose number 1, he went to the emergency room for what I'm going to show you. He was prescribed cephalexin, and with 8 hours, she discussed with the patient to hold the cephalexin and monitor, and his arm swelling reduced significantly in 24 hours. But let's see what that meant. So he's given permission to share these. At 12 hours on the left, he had a big red arm, and that's bigger than the other pictures I showed you. At 24 hours, he looks like he has a welt on his arm surrounded by erythema. By 36 hours, he looks like he has a full-blown cellulitis, and you can see the rings where he was very smart in monitoring how the induration and erythema was progressing. But by 6 days after, he did have all this shrinkage, but this one area of pigmentation on his arm. And so that's something to think about in terms of, as you can see, CDC recommends against using topical steroids or antihistamines. Ice packs are good in oral antihistamines because he also said it was itchy. And because of this potential marking that would say, oh, you must be someone who got monkeypox vaccine, other sites of administration of this intradermal dosing are recommended, either in the upper back below the scapula or very high up on the deltoid. So what I hope I've taken you through a quick run through of how we got to this MVA Bavaria Nordic. It's actually been around for much longer than people think. We have a lot more data about this vaccine than people have, that most people know about. It's had quite a development history. It has similarities and differences with the live replicating vaccine. They both can be used against monkeypox and smallpox, but because ACAM2000 is only on label and only licensed for smallpox, as opposed to Hynes, which was licensed for both smallpox and monkeypox, it would have to be used under an EUA. And then I think you understand why there are some provisions for Hynes, why it has to be used under an EUA. And then the efficacy and immunogenicity of Hynes really isn't really bad, even after 1 dose, but 2 doses are what's needed to give you the maximal equivalence to the ACAM2000. And so then we also looked at some of the possible adverse effects for the intradermal Hynes vaccine. So that's about it. And I'm happy to hear any questions that came in while I was giving this nonstop, sounds like a diatribe, but thank you for listening.
Wonderful. Thank you so much. And I don't have any questions yet, but I'm assuming people are probably about to start typing. So I will ask a quick question. And also thank you for giving this entire history, because I agree with you that it is very important to know the entirety of a history of vaccine development, especially when it is, you know, a vaccine for a new outbreak that, you know,
Yes.
people might have just heard about, but
Mm-hmm.
the vaccine itself isn't new at all. So thank you for giving
that's the reason why I went into all the history, to understand why we're not using ACAM2000, but also why really MVA Bavaria Nordic is not really a new vaccine. It's been in development for over half a century.
You had mentioned in a previous lecture about instructing patients that get an intradermal vaccine if it's itchy, not to scratch it.
Yes.
And I want to make sure
If-
that gets said.
Yes. If you get an intradermal vaccination, you get a bleb. Very often people go, it itches, and they take the gauze or whatever you place over it and they press down on the vaccine. You will convert an intradermal vaccination to a subcutaneous vaccination if you press it down and make the bleb go away. So you'll be giving them a 20% dose. So it's an inadequate dose. So you have to tell them, don't press on this. Yes, thank you for reminding me, Barbara.
Oh yeah, no, I that was my first time hearing that, and I think that's so important for people to know as well.
So, and also, if any of you are still giving PPDs for detection of tuberculosis MTB exposures, if people do that and press their bleb down, if they have a negative, it could be quite a false negative.
So the take home is don't push any intradermal thing, don't press on it, leave it alone.
Yes, correct. So do we have?
I am not seeing any questions. I will start doing my slow talk to see if someone is still putting something in. Lots of thank yous in the chat.
You're welcome.
And if we have no further questions, then I do want to thank you so much, Dr. Hynes. This is, again, like just amazing. And I want to thank everyone for attending. And please, if you have other colleagues that you think would benefit from this information, even if they're not going to get CE credit, direct them towards the Maryland Department of Health website where they can see the recording. Again, fascinating. So I thank you so much for your time and expertise and wisdom. And everyone have a wonderful night.
Yeah, and if people come up with questions and they shoot them to you, Barbara, or you, Jean, or you, Bambi, you can let me know. I'm happy to answer them to the best of my knowledge. I thank all of you for staying these few extra minutes. Much appreciated.
Wonderful. Thank you, and everyone have a great night.